Hello everyone, myself Jyotsna Priyam and I am PhD research scholar in Department of Biotechnology, National Institute of Technology, Warangal, India. Here I am going to present my work whose title is Carbon-Based Nanomaterials in Renal Cancer. Renal cell carcinoma accounts for 2% of all cancers globally. The rate of mortality is very high in metastatic renal cell carcinoma. Traditional methods of treatment like ultrasound and chemotherapy have their limitations as per reports. Like metastatic renal cell carcinoma is radioresistant and systemic chemotherapy is also not effective in some cases of advanced renal cell carcinoma. The limitations of traditional therapy methods necessitate the design of advanced therapeutic strategies of renal cell carcinoma, and carbon nanomaterial is one of them. Carbon-based nanomaterials are formed by carbon atoms organized in a specific nanoscale configuration. They are formed by covalent bonds between carbon atoms. Examples of carbon nanomaterials are single-walled carbon nanomaterial, multi-walled carbon nanomaterial, nanodiamonds, nanohorns, graphene, graphene oxide, and so more. Their unique characteristics like size and chirality make them suitable for biomedical applications. One of their applications is as therapeutic carrier in renal cancer. This diagram is divided into 4 parts. This part is showing examples of different types of carbon nanomaterials like single-wall carbon nanomaterials, multi-wall carbon nanomaterials, graphene oxides, and fullenes. Again, this part is showing chemical functionalization of carbon nanomaterials with peptide, DNA, and antibody. Chemical functionalization involves changes in size and configuration of carbon nanomaterials as well as alteration in its surface to target kidney cancer cells. This can be done by addition or removal of charged groups or ions by covalent or non-covalent bond, oxidation or halogenation, and cycloaddition. This part of diagram is showing efficiency of carbon nanomaterial in enhancing drug delivery to renal cancer cell. This bar graph is reflecting higher percentage of apoptosis in renal cancer cell when drug delivery by carbon nanomaterials takes place. Besides drug delivery, carbon nanomaterial can also be used as gene delivery and peptide delivery. Gene delivery can be done by coupling gene to the surface of carbon nanomaterials or encapsulating them inside carbon nanomaterials to shield them from degradation. This reason is reflecting the theranostic methods of renal cell carcinoma treatment. One of them is hyperthermia. Hyperthermia is a thermal therapy in which cancerous tissues are removed by 41 to 47 degrees centigrade temperature while normal tissues remain unaffected. Radio frequency energy and laser beam are used as energy sources along with carbon nanomaterials to eradicate cancerous cells. Similarly, reactive oxygen species are created which are capable of killing renal cell carcinoma during photodynamic therapy using photosensitizing agents. Carbon nanomaterials absorb a specific wavelength of light, making them appropriate for use as photosensitizers in photodynamic therapy. We also studied effects of physical characteristics like toxicity, enzymatic degradation, biocompatibility, and biocorona in renal cancer treatment with carbon nanotubes as shown in this diagram. These are the limitations of carbon nanomaterials. Starting with toxicity, cells are vulnerable to damage from metal impurities added during production of carbon nanomaterials. Besides, length and diameter, if increased than the optimal value, can also cause toxicity. Another physical characteristic is enzymatic degradation. All enzymes are proteins and they break down into biomolecules inside the body. Carbon nanomaterials that can tolerate enzymatic degradation are more effective at targeting drug for renal cancer treatment. Similarly, loss of biocompatibility also increases the risk of side effects, and carbon nanomaterials can evoke immune responses inside the body. Biocorona is a phenomena when carbon nanomaterials are placed into a biological environment, their surface becomes coated with a variety of biomolecules forming biocorona. It can act as a barrier in drug delivery via carbon nanomaterials. As shown in this diagram, the x-axis of this graph is temperature axis and the y-axis is percentage decrease in mass. This is a graphical representation of biocorona in carbon nanotubes. there is a sharp decline in percentage of decrease in mass as temperature rises in case of biocoronated carbon nanotubes, which is carbon nanotube plus serum, in comparison to the carbon nanotubes alone. Additionally, when carbon nanomaterials react with tumor microenvironment, it causes modulation of immune response, regulation of tumor hypoxia. They also interacted with macrophages and endothelial cells of tumor microenvironment. As a concluding remark, we can say that carbon nanomaterials hold significant potential for enhancing renal cancer therapy, and they can improve chemotherapeutic drug delivery to renal cancer cells. Thank you.